So this session is going to look at the host responses to viruses. But of course, there's a lot of common features when we're thinking about the response to other infections, such as bacteria. So really what this session is going to be about, it's an overview or a summary of the immune system, how the immune system functions against microorganisms, with a slight focus on viruses. So first of all, we'll look at the innate immune response to infections before going on to look at the adaptive immune response to infections. So we're going to separate it into those two little parts. And so if you're looking at the course of a typical acute viral infection, which is what this image is showing, on the y-axis you can see the virus growth, so the amount of virus that there is, and there on the x-axis is the duration or duration of the infection. So you can see when a virus enters your body, it has to enter cells before it's going to do anything itself, before it can start replicating. But of course, very early on, the innate defenses start kicking off. Okay, so the red is just showing the amount of virus. So you can see the amount of virus that's entered the cells, it's starting to replicate within the cells, and your innate defenses are kicked off. You then start over time induce your adaptive response. The adaptive response continues to fight the infection, and there you can see the virus growth disappearing and the virus being cleared. And then you form your memory cells, your memory B cells, etc., so that you're able to fight the same infection much quicker next time. And of course, you do actually need a threshold level, and that's what this little area is showing. You need a threshold amount of virus to stimulate the adaptive immune response. So what are some of the barriers to infection? So a lot of this has already been covered, and a lot of it is very simple. So of course, the skin itself is a physical barrier to infection, to microorganisms. It stops a large number of microorganisms from entering. But of course, there are receptors on multiple cell types that allows them to get in, that allows viruses to get in. So receptors are there, so viruses bind to receptors for entry. And we then have mucus, of course. So there's lots of mucus that is present. And mucus is great because it acts as a kind of a fake receptor. So some viruses will bind to glycoproteins present within the mucus, thinking that they're about to get into a cell, but actually all they've done is getting bound to a fake receptor. So your mucus has a very important role. Of course, things like lower heat pH are very important because gastric secretions inactivate most viruses. There are notable exceptions, such as rotavirus. So the rotavirus is a virus that mainly affects young children with nausea, vomiting, and diarrhea. And it has multiple membranes. They're protein membranes around it. They're made up of protein. So protein shells is a better phrase, that surrounds the nucleic acid within it. And that helps to protect it from the low pH within the stomach. Of course, you also have ciliated epithelium, which help to remove some of the respiratory viruses, for example. And that's just what this image is showing. It's just showing some of those epithelia that will help to remove viruses through the elevator system. And of course, these barriers are the first line of defense against microorganisms. And the host will be resistant to a virus if it doesn't have the correct receptor or machinery required for its replication. So, for example, humans are resistant to all plant and most other animal viruses because they don't have the correct receptor to allow the virus to get into the cell. Because remember, viruses are a type of parasite, and actually they do have to get into the cell. They use the host cell machinery to replicate. So if we look at the immune responses that kick in following an infection, so again, if we're looking at the y-axis, you can see this is the antibody titer of the antibody titer of virus rather. You can see the blue is the amount of virus that's in your circulation. You can see fever kicks in as the virus gets to much higher levels, and the x-axis is obviously days after virus administration. And you can see early on you've got your non-specific host defenses. They kick in immediately after infection, whereas those specific defenses take days to weeks to mount and respond to a viral infection. So, as we've already talked about, the innate immune response is non-specific. It's that first line of defense against infection. It consists of a variety of soluble factors, such as complement proteins, and other diverse cellular components, including your NK cells, that's your natural killer cells. The adaptive immune response, as we know, is more specific, but it's slower to develop, but manifests as increased specificity and also of memory. And it consists of antibodies, your B cells, your CD4 positive and your CD8 positive T lymphocytes. So those are your helper and killer T lymphocytes. And you can see that over time, antibodies start to kick off. And I just want you to quickly say to yourself, what is the first antibody type that kicks in following an infection? So is it A, D, E, G, or M? So it's AGM. IgM is the first antibody that kicks in following an infection. And then that rises quickly, then drops, and then IgG takes over. So if we're looking at some of these innate immune responses, we've got the interferons. So these are a group of proteins that help defend against viral infections. And we will go through each of these in turn in a second. We've got our natural killer cells. These are cells that non-specifically lyse and destroy virus-infected cells as well as cancer cells. So our bodies produce a vast number of cancer cells every day by accident, and we have methods of killing them. And one of those is through our NK cells, our natural killer cells. We have the complement system. So this consists of a series of plasma proteins that, when activated, help to destroy foreign cells. And we've got phagocytosis. So again, these are your macrophages, which are specific to tissues, your monocytes, which are floating around the blood, your dendritic cells, etc. And they help to engulf viruses and other pathogens. So we're going to first of all focus on interferons. And we're really, like I said before, this is only a summary of how the immune system functions, and we're targeting viruses in particular. But like I said, a lot of this, not so much interferons, but a lot of this you can relate to other microorganisms. So interferons, they interfere with the replication of the same or unrelated viruses in other host cells. So how does that actually work? Well, a virus gets into a cell, so it binds to a receptor, gets in, starts replicating and doing its job. The cell then releases interferon. Okay, an interferon isn't released in response to bacteria, for example. The interferon binds with receptors on uninvaded cells. And now that these uninvaded cells are being stimulated, they produce enzymes capable of breaking down viral messenger RNA. So remember, all viruses have to make messenger RNA, whether they're an RNA virus or a DNA virus, they have to make viral messenger RNA because they don't have ribosomes. And so they make viral messenger RNA. That viral messenger RNA binds to the host ribosomes, and they go on to make their proteins. So like I said, the uninvaded cells are now induced or stimulated to produce enzymes that break down viral messenger RNA. And now, so the virus from this other cell enters this previously uninvaded cell that has been activated with interferon, and these virus-blocking enzymes have been activated. So the virus is now unable to multiply in the newly invaded cells. So those cells can go on and live a happy, jolly life. And we don't have any new viral proteins being made. Unfortunately, some viruses express proteins that interfere with the interferons. For example, HPV blocks some of the downstream signaling after it binds to its receptors. Others produce interferon homologs that bind to the receptor, while others secrete fake receptors that bind to interferon itself. So viruses are very smart. They come up with mechanisms to actually evade or inhibit interferon from doing its job. But interferons are used in therapy against some viruses. So interferons can potentially be used in Hep B and Hep C infections, for example. The next one, nice, simple, NK cells, your natural killer cells. These destroy virus-infected cells by basically lysing their membranes. So a virus infects a cell, the NK cells, which have been activated by interferons, they arrive nice and early, and they recognize stress cells. So therefore, they can act much faster in destroying infected cells than the more specific CD8 T cells that bind to your MHC, your major histocompatibility complex. So they're just basically recognizing these stressed cells, these infected cells. So they go on to bind the virally infected cell. They inject some cytoplasmic granules of proteins, so perforin and granzyme, and those two things together basically cause holes to form in the cell membrane. And remember, it's a virally infected cell. And so therefore the cell dies by apoptosis, so programmed cell death, which prevents the growth and spread of the virus, because apoptosis, as well as causing programmed cell death, also causes degradation in a controlled way of the virus, unlike cell lysis, which just releases the whole virus and lots of it. So if a cell dies by necrosis, just death, it just releases lots of virus. If it dies in a controlled way, it helps to prevent the growth and spread of virus. We've then got the complement system. So this is a cascade of reactions that help or complement, hence its name, the ability of the immune system to clear pathogens from an organism. And so at the end of the whole complex, so there are a number of small proteins found in the blood, and together they form what's called a membrane attack complex in the membranes. And that basically forms this like a hole in the cell membrane, and basically the cell begins to die because it can no longer control its intracellular environment. And so only enveloped viruses, so those are ones that have got a lipid envelope, or virally infected cells are adversely affected by complement. Because that, like I said, a lipid bilayer is required. There are some viruses, an example I gave you earlier was rotavirus, that doesn't have a lipid envelope surrounding it, helping to protect it. And so if we're thinking about complement, we have the antibody-dependent pathway. So this is where your complement proteins bind to an antibody-antigen complex. So here is your antigen, your virus, if you like. Here is your antibody. That then activates the complement system, and we get virolysis, so degradation, lysis of the virus. So it basically causes the virus membrane to lyse open. So now you can see why it requires a viral lipid membrane or a lipid bilayer, because the antibody binds to the virus, and then that activates complement, and complement basically drills a hole in the lipid membrane of the virus, killing that virus. You've got the antibody-independent method, and that's where the complement binds to virally infected cells, and then that puts that complement-mediated hole into the plasma membrane of the infected cell, and you get lysis of the infected cell. So you've got the antibody-dependent and the antibody-independent methods. Finally, last little one, you've got your phagocytes, and really very simply, these are your big cells that they go around and engulf and ingest foreign materials such as viruses before degrading them within the phagosome, which is similar to a lysosome in another cell. So they basically degrade the viruses. Okay, so let's have a little look and see how you're doing. So have a look at these questions, press pause, and then we'll go through them. Which of the following are correct regarding the innate immune response? Natural killer cells can be activated by interferon. That is correct. Natural killer cells produce proteins such as perforin and granzyme that can kill both enveloped and non-enveloped viruses. That is incorrect. Holes form in the membranes, remember, in the lipid bilayer. Complement activation causes pores to form, which kill both enveloped and non-enveloped viruses. That is incorrect. The holes form in the membrane of enveloped viruses. They can't form in protein complexes surrounding viruses. Question 2. Which of the following are correct when interferon from one cell attaches to a second cell? Virus can enter the second cell. That is correct. The virus can replicate within the second cell, but cannot be released. That is incorrect. It can't replicate because it can't make any new proteins because the messenger RNA is being damaged. Number 3, the second cell now makes interferon and saves itself. That is incorrect because it's already been stimulated by interferon, and that interferon that stimulated it to make enzymes that degrade the viral messenger RNA, so it doesn't need to make interferon. The recipient cell makes enzymes that degrade viral messenger RNA and prevent viral protein synthesis. That is correct. Remember, it makes enzymes that degrade messenger RNA and therefore prevent viral protein synthesis and therefore prevent it from replicating. Oh, answers the next one. Viral replication takes place as usual. That is obviously incorrect. So the second part we're going to look at is the adaptive immune response to infections, again with a focus on viruses, but it's very relevant for a multiple of microorganisms, so it's a good little review session for you. So of course, in the adaptive immune response, we have both the humoral and the cell-mediated response, and the humoral is all about the B cells. So you have your specific B cells, which can proliferate and produce antibodies against specific viral proteins. And these antibodies can block the infectivity of the virus. This is termed neutralization. But of course, sometimes an antibody response is in itself pathogenic. For example, immune complexes are thought to be responsible for causing the rash in rubella infections. So as well as the humoral, you've got the cell-mediated response, and this is the cellular response, and that helps to kill the virally infected cells. So in other words, the antibodies are important in preventing viral infections, while cell-mediated response helps to eliminate infected cells. So there are specific key roles for each part of the adaptive immune response. So if we're thinking about the humoral response, so we're thinking about antibodies, we're also thinking about neutralization. I told you what neutralization was in the last slide. At what stages do you think antibodies may neutralize viruses? So stop them doing their job. Have a look at the diagram. It'll help you think about it. Where are antibodies helping to neutralize a virus? So stop it getting in, doing its job. So you can stop it from binding to its receptor. So an antibody that you can see there binds to a virus and stops it binding to its receptor. It can also block the virus uptake, which is the second one. So a lot of them get taken up through receptor-mediated endocytosis, for example. So taken into the cell via a receptor and into an intracellular compartment. And so you can stop the machinery from working and therefore getting endocytosed into the cell. Even if it gets taken up into the cell, you can prevent it from being uncoated and the release of its genome, whether that genome is DNA or RNA, doesn't really matter too much, but it needs to fall apart. The protein membrane, the protein layer needs to be removed, or the membrane layer needs to be removed, so you can start to block it. You can do that by stabilizing the capsid, and the capsid is the protein layer that surrounds the nucleic acid as a general rule. Or you can prevent fusion. That's where the lipid membrane may fuse. So the lipid membrane of a virus may fuse with the lipid membrane of this intracellular compartment, whether it's a lysosome or an endosome, for example. And you can also lead to virus aggregation, and that's shown in this top image here. So the virus, basically, the antibodies join lots of viruses together, leading to virus aggregation, and it can no longer infect cells, so it's been neutralized. So many enveloped viruses are lysed when antiviral antibodies and complement disrupt the membrane, and we talked about that already in the previous section. So what are the types of antibody that we're most concerned about? So of course we have our IgM. This is the first immunoglobulin to respond to a viral or other type of infection. And so if IgM is found in plasma samples, it indicates a recent or current infection, as it only lasts generally a couple of weeks. IgG, as you'll know, is the most abundant antibody, making up about 75% of the plasma immunoglobulins. It's found in plasma samples and it's indicative of a previous or past infection. It's long-lived and therefore helps to provide years of protection. And that's what you're trying to stimulate, as well as memory cells. You're stimulating IgG to sit around as part of a vaccination protocol, for example. So here's a typical response to a viral infection or to a vaccine. And so when you get your first, so this is the amount of antibody on the y-axis and then time on the x-axis. You stimulate with the antigen. So that might be a vaccine, for example, or it might be an infection. And you can see that IgM kicks in first, but it doesn't last that long. Its levels start to reduce quite rapidly. IgG kicks in a little bit later. It gets a bit higher and it stays up a little bit longer before decreasing. And you can see the total antibody response that's there. If you then give a second antibody response, so you give a second vaccination, for example, these are typical responses. Give a second one, you can see IgM kicks off again. You can now see that IgG kicks off faster and it goes higher and it remains higher for longer. And that's because, of course, you formed your memory cells, your memory B cells, which are then able to be stimulated very fast and produce your immunoglobulin. Okay, some more questions for you. So press pause, have a think. The immune response that is directed at discrete molecules on pathogens or viruses is termed the innate response. True or false? That is incorrect. Adaptive response. That is correct. Phagocyte response. Incorrect. And the passive response. Also incorrect. Which of the following are correct regarding a viral infection? IgG is the first antibody produced following virus exposure. That is incorrect. It is IgM. IgM confers long-term resistance to viral infections. That is incorrect. It is IgG. Number 3, IgG is produced at high levels on second stimulation. That is correct. IgM, number 4, IgM is the most abundant antibody in the blood. That is incorrect. It's IgG. Number 5, IgM contains 5 immunoglobulin molecules joined together. That is correct. So we're looking again, like we said, we're looking at the adaptive immune response, but now we're going to look at the cell-mediated response, and specifically we're going to talk about T cells. So what's happening with T cells is they can recognize cells that contain virus-derived peptides displayed on the outside of the infected cell. So what happens? A virus, so we're just looking at this little section that's in the red box. We're looking at viruses infected the cell. Some of the viral proteins get degraded, and this little thing that you can see in this is a proteasome, what's called a proteasome. So the virus has infected the cell. Some viral proteins are degraded within the proteasome, within the cell. These viral proteins are transported to the endoplasmic reticulum via a TAP. It's called a TAP transporter. Doesn't matter too much about what it is, but they go to the endoplasmic reticulum, binds to the MHC class 1 molecules, and all cells can will express MHC class 1 molecules except for your red blood cells. And these will then pass up through the secretory or the exocytic pathway, and then they will pass onto the plasma membrane, where they'll be expressed on the plasma membrane. So basically, you've now got part of this viral peptide or an antigen being presented ready for other cells to come in and be stimulated it or detect it. And so this is now detected by your cytotoxic T lymphocytes, your CDLs. Okay, these are the ones that are CD8 positive. So these are called your cytotoxic T lymphocytes, and they go on to kill the cell. So like a cell, in this case, your virus has infected a cell. Part of the viral proteins have been broken down. Those viral proteins are sent to the surface of the cell. This is detected by your cytotoxic T cells, which come in and kill the infected cell. Nice, simple, and it's happening in pretty much all cells apart from red blood cells. So like I've said, what's actually happening is when your CD8 positive cells, these are your cytotoxic T cells, when these bind to the virus-derived peptides, you've got a little complex going on. You've got your CD8, you've got various receptors. It doesn't really matter too much. But here's your cytotoxic T cell. There's your virus-infected cell, and there's your little peptide on the surface. So then the T lymphocyte releases perforin and granzyme are released into these infected cells, and the cells undergo apoptosis and die in a controlled way. And most T cells die, but some do remain to form memory T cells. But what about the helper T cells? So these are the CD4 positive cells. So your B cells can bind to virus particles. So if we're looking at this image, your viruses, if you're looking at this image, the B cells have got their antibodies on the surface. The viruses can bind to those antibodies to form an antigen antibody complex. So we've now got a B cell attached to an antigen, or attached to a virus in this case. And this is where we hit the exogenous pathway. So the B cells can now internalize through the exogenous pathway that bound virus. So it now internalizes that virus into itself. The viral proteins are then digested and presented onto the surface to CD4 positive T helper cells. So again, they're broken down and presented onto the surface of the cell. And so what happens now? So now the T helper cells are being stimulated because of these B cell binding, and the T helper cells secrete a variety of things. They secrete cytokines, which can activate many aspects of the immune system. So they can activate your B and T cells, and they can activate your macrophages. They stimulate the growth of immune cells, such as your memory, your T helper, and your cytotoxic T cells. They attract immune cells to the infection site. For example, your macrophages and your T cytotoxic T cells. But remember, of course, your T helper cells also respond to viral antigens that are presented on macrophages. So macrophages that are going around and gobbling up viruses and chomping that virus up, they are also stimulating our helper cells. So the T helper cells are basically helping to stimulate the immune system, where the cytotoxic T cells are there to kill infected cells. So another little question for you. You've got a lot of questions today. Cytotoxic T cells interact with virus-derived peptide on infected cells and kill infected cells by which of the following mechanisms? Produce antibodies. That's incorrect. Produce cytokines which stimulate macrophages to kill the cell. That's incorrect. They produce perforin and granzyme which induce apoptosis. That is correct. They stimulate B cells which kill infected cells. That is also incorrect. Number 2, which of the following are functions of T helper cells? Number 1, activation of many cells of the immune system. Of course, that's correct. We've just done it. They release perforin and granzyme into the infected cells, leading to their death by apoptosis. That is incorrect. Remember, it's the cytotoxic T cells, the CD8 positive cells that do that. Do they attract immune cells to the site of infection? Yes, they do. Do they stimulate the growth of immune cells? Yes, they do. So really, what are some of the key points about the adaptive immune response? Well, the key major players are your B and your T cells. So your B/plasma cells for antibodies and your T cells in terms of helper and cytotoxic. The cytokines play an important role in activating or communicating between B and T cells. Repeated exposure to the same pathogens results in memory B and T cells. And the adaptive immune response takes some time to mount. Hence, the innate immune response provides immediate action against any invading pathogen before the adaptive immune response helps to eliminate that pathogen.